Nanobots are machines whose components are at the nanoscale and can be designed in such a way that they have the ability to move autonomously in fluids. Although they are still in the research and development phase, significant advances are being made toward the use of nanorobots in biomedicine. Their applications are varied, from the identification of tumor cells to the release of drugs in specific locations. Nanorobots powered by catalytic enzymes are among the most promising systems because they are fully biocompatible and can make use of fuels already available in the body for their propulsion. However, understanding the collective behavior of these nanorobots is essential to advance towards their use in clinical practice. Now, in a new study published in the journal Science Robotics, researchers led by ICRIA research professor Samuel Sanchez and his team, Smart Nanobio Devices, at the Institute for Bioengineering of Catalonia, together with a group radiochemistry and nuclear imaging lab from SIC Biomagune led by Jordi Lop and the Universitat Autònoma de Barcelona, have managed to observe in vivo the collective behavior of a large number of autonomous nanorobots inside the bladder of living mice using radioactive isotope labeling. The fact of having been able to see how nanorobots move together, like a swarm, and of following them within a living organism, is important, since millions of them are needed to treat specific pathologies. Nanorobots can be monitored in vivo through positron emission tomography, a highly sensitive, non-invasive technique used in the biomedical environment. To do this, the researchers first carried out in vitro experiments, monitoring the nanorobots through optical microscopy and positron emission tomography. Both techniques allowed them to observe how the nanoparticles mixed with the fluids and were capable of migrating, collectively, following complex paths. The nanorobots were then administered intravenously to mice and, finally, introduced into the bladders of these animals. Since nanorobots are coated with an enzyme called urus, which uses the urea from urine as a fuel, they swim collectively, inducing fluid flows inside the bladder. This study demonstrates the high efficiency of millions of nanoscopic devices to move in a coordinated way in both in vitro and in vivo environments, a fact that constitutes a fundamental advance in the race of nanorobots to become the key players in highly precise therapies and treatments. Future applications in medicine. It has also been demonstrated that the movement of these devices can be monitored using imaging techniques that can be applied to the in vivo environment. In other words, they can be applied in test animals and offer the potential for transfer. This is the first time that we are able to directly visualize the active diffusion of biocompatible nanorobots within biological fluids in vivo. The possibility to monitor their activity within the body and the fact that they display a more homogeneous distribution could revolutionize the way we understand nanoparticle-based drug delivery and diagnostic. Nanobot swarms could be especially useful in viscous media, where drug diffusion is often limited by poor vascularization, such as in the gastrointestinal tract, the eye, or the joints. In fact, as different enzymes can be incorporated into the tiny motors, nanorobots could be tailored according to the part within the organism, adapting the device to the accessible fuel in the environment.